Hello friends, welcome back to my channel. Okay, so that means we can say that HIV part 1. Sorry, it should be said that HIV part 1. Now see, we'll just start the series of the videos in terms of the HIV and we'll complete within the 4 parts. Okay, first today we'll talk about the normal basic organs, so the structures of the HIV. Okay, and on the the complex, or you can say in details the pathophysiology. We'll talk about all these things in details in the subsequent parts. Okay. Now, whenever we are talking in terms of the HIV, as we know, there is the human immunodeficiency virus, and when we get this type of the problem, whatever the problem we'll see from the name, it's easy to understand. It will cause the immune deficiency, so we'll get the immune suppressions. So whenever we get the immune suppressions, at that time we'll get the various kinds of opportunistic infections that will be increased. So as a result, we can say that whatever the problem we'll see over here, as we will get the immune suppression, immune suppressions. As a result, we will get what the opportunistic infections, chances, the opportunistic infections that will be increased over here. Now, along with that, with the main problem we'll see over here that here we'll get also the CNS or neurological disorders. Okay. Now, cause the immune suppressions, how they are doing these things, we'll talk about all these things within a minute. And as a result, we'll get the chances for the opportunistic infections that will be increased. Disorders. Okay. Now, as we know, that's the HIV. It's a what? RNA retrovirus, right? It's a RNA retrovirus. RNA retrovirus. Now we'll talk about these terms. Okay, the retrovirus. Now, what do you know? Normally, if we see under normal physiological circumstances, like in the other animals, including human beings also, as we know, first we'll get the DNA. From DNA we get what? The RNA. From RNA we get the protein, and this protein may be the structural protein, or you can get the functional protein. The structural that will develop the structures, functional in terms of the different kinds of enzymes and the regulatory proteins. Okay, all these things will get. And here we'll get also from DNA to DNA. That's the auto replications. These are transcriptions. These are translations. Right? These whole things, as we see, this thing is known as central dogma. But what about the RNA retrovirus? Whatever the changes we'll get in these cases. See, in these cases we'll get the productions of the DNA from RNA. From RNA to DNA we'll get these portions. From RNA to DNA we'll get RNA to DNA. Now, whenever you're getting the productions of DNA from RNA, that's what there is the reverse process. Okay, that's what is known as reverse transcriptions. What do you know in normally in the transcriptions process? What do you know? Try to understand from the DNA to RNA we get. This is transcriptions. But here we're getting from RNA to DNA. So that's the reverse transcription. So you can write it over here. This is the reverse transcriptions, right? Reverse transcription. Now, as we know, that's the reverse transcription. Just to convert the from RNA to DNA, we need to use an enzyme. We need to use an enzyme. What is the name of the enzyme? That is the reverse transcriptase, right? The reverse transcriptase. Now, the reverse transcriptase enzyme that will be carried by the HIV virus only. So, the enzyme that will be present within the HIV. So, we'll get the reverse transcriptase enzyme. The reverse transcriptase enzymes. Okay. Now, ultimately we'll get what? From the RNA we'll get the DNA. Okay. And from DNA we'll get the productions of the mRNA. Okay, let's use this color. From DNA we'll get the messenger RNA. Okay. From the DNA we'll get what? The messenger RNA. From the messenger RNA we'll get what? The protein. And also we'll get the genomic RNA. Genomic RNA. And these two, this protein and the genomic RNA, they will have the capacity to produce again the new viruses, right? From here we'll get the new virus. This is the normal thing as we know. Okay. Now we'll talk about basically we'll focus on the structures of the HIV virus. Now, as you know that when you will get the immune suppressions as a result, This type of thing we'll see that will lead to ultimately what the AIDS, right? Acquired immunodeficiency syndrome. Now, as we know that the HIV virus, okay, let's use this one. The HIV virus would like to bind with, like to bind with CD4 positive cells, right? CD4 positive cells. Now, in which cases we will get the CD4 positive? In which cells we will see the CD4 receptor is present? In which cells we will get the CD4 receptor is present? Now, where we will get the CD4 positive? In terms of, as you know, the T helper cells. All right, we will get, the monocytes. So whenever we are getting in terms of the monocytes within the circulations, whenever they will come out of the circulation, they will be known as macrophages. So you can write it over here also in terms of also the macrophage. Now whenever we get this type of cells, so under the skin, this type of the cells whenever we get under the skin, that is known as what? Dendritic cell, right? Or in the better word we can say the Langerhans cells. Langerhans cells. Or we can say that in terms of the, within the or in the central systems or the microglia. So that will be modified over here. For example, within these cells we can get what? Also we can get in terms of the follicular cells within the spleen, okay? CD4 positive receptors. Now this HIV virus, they would like to attack or get bind along with the CD4 positive cells. In terms of the CD4 positive cells, we can get the T helper cells, we can get the monocytes or the macrophages, we can get the Langerhans, we can get the microglia, okay? Now, at first we need to know what type of the structures we will see in terms of the HIV. Okay, first we will see the HIV structures, and then we will go to the details. Okay. Now see, as we know, there is the HIV. There is the what? The lipid bilayer. Which is within the HIV, we will get the lipid bilayer. How the HIV is getting the lipid bilayer? Is the normal cell? Is the normal cell? Okay, and whenever the HIV virus, they will come over here. Okay, whenever they will be within the cells, and whenever they will be out of the cell, at that time what they will do, they will steal the lipid bilayer membrane from the host cell, right? They will steal this membrane. That means ultimately we will get the structures that will be, for example, along with that we will also get the lipid bilayer membrane in the HIV. Okay, this part that will be stored by the HIV. So from the host cell, the HIV, they will take the lipid bilayer membrane, and after that they will come out from the cell. Okay. Now we will talk about details about the HIV structures. Now see, as we know, the HIV structures, as I said you, they are the enveloped structures because they are having the lipid bilayer membrane. So let's talk about all these things right now. For that thing, I will draw a little bit. Okay. The lipid bilayer membrane. Now within the lipid bilayer membrane of the HIV, we will get a transmembrane protein over here. We will get a transmembrane protein over here. Okay, transmembrane protein. Now this transmembrane protein, that is known as GP41. This is known as GP41. Now along with it we will get other structures, another proteins along with it will get bind. We will get another binding of the another protein along with the GP41 over here. This is known as GP120. This is known as GP120. GP means glycoprotein. Okay. Now GP41 and GP120, they together. If we can say in terms of the GP160, no, we can say GP160, not 161. Okay, we have to say in terms of the GP160. So if we just do the additions of them, we can say in terms of the GP160 only. Now, this is the site. This GP120, they will bind. They are having the capacity to bind with target cell receptor, right? Target cell or receptor, target cell receptors. in terms of the CD4 positive cells. Now, how they are getting bound along with these structures, let's talk about all these things. In the same way, we'll get also over here the GP, in the same way we'll get GP41 over here, all over the membrane. We'll get the GP41. Here we'll get also the GP41. Here we'll also get the GP41. Along with those, we'll get also the protein GP120, right? GP120, GP41. GP41. Anyway, I just told up to this much. Okay. Now, whenever you're getting the GP41, as I said you, whatever the thing will get. Okay, let's show these things with a small part. I'm just considering this whole thing in this way. Okay. This is the HIV virus along with the 2 getting. This GP41. Let's write it clearly. GP41, and along with it will get what? Along with it we'll get the GP120, right? We'll get the GP120. Now, as I said, we have to get on terms of the what we have to get in terms of the CD4 positive cells. Now, for example, I am just giving the example with the T cells. Okay. Okay, let's use the different color for it, guys. But which color should I use? Okay, let's use the red color. For example, is the CD4 positive T helper cells. Now, as I said, there will be the presence of what? There will be the presence of CD4 receptor, right? CD4 receptor. So let me show the CD4 receptor with this color. This is the CD4 receptors. CD4 or receptor. Okay. Now try to understand from this figure. As I said you, the GP120, glycoprotein 120, that will get bound along with what? Along with the receptors, that means the target cell receptors, they are also CD4 receptors. Now whenever it will get bound along with the CD4 receptors, try to understand from here, we'll get what? Some signals that will raise within this GP120, and within the GP120 we'll get another type of the structures. The conformational changes will get where within the GP120. Now whatever the conformational changes will get over here, it will strike another co-receptors or from here, or you can say the stimulated from these regions, and that bound another structure that is present where within the CD4 positive cells. Okay, so let me repeat once again. Whenever the GP120, let me write it over here, GP120. So whenever the GP120 that will attach along with the CD4 receptors, at that time, see, this GP120, there within the GP120 will get a conformational changes. Whatever the changes, it will extend another co-stimulator or the co-receptor, we can say the co-stimulator, that will go towards the CD4 or the CD4 positive T cells, for example, over here. Okay. Now, whatever the thing will get over here, let's talk about, let's consider the CD4 positive. I'm just taking the macrophage, then I'll say it. It is macrophage. The T cells, I'll just show it over here. The T cells, for example, I'm just considering this is the CD4 positive macrophage. Now, okay, basically this receptor that is getting over here within the on the macrophage. These are the receptors for the different kinds of the what? Leukotrienes, or we can say the chemokines. So these are the receptors. Normally there is a present for the different kinds of the what? Interleukins, we can say also over here, we can say the chemokines. But whenever the HIV is coming over here, this HIV that will utilize this receptor for its own purpose. Normally what? Normally this receptor, you can write it over here, that is known as CCR5. Chemokine receptor 5. Now see, normally the CCR5 that is present, what? The CCR5 that is present where within the macrophage for the different kinds of the what? Chemokines. But whenever the HIV that will take place within the cells, where the HIV that will utilize this CCR5 receptor for its own purpose. Now this CCR5, whenever they will attack it over, whenever they will get attached along with these regions. So whenever they will get bound along with this structure, the co-stimulators over here, and the same will also get another, same thing will get over here. It will be activated. Now in the same way, try to understand, we will get whenever this all these things, see this GP120 that will get bound totally along with the CD4 receptors or the CD4 positive macrophage cells over here. At that time we'll get another signal, so the ultimate signal that will reach from where, which is the ultimate signal that will reach from the, okay, let's use a different color for it guys. From here we'll get the ultimate signal that will reach in this regions, and whenever these signals from the, from where, from the GP41, from the GP41 we'll get the ultimate signal that will reach where, within the membrane of the macrophage, and the signal that will reach, or we can say the the stimulator that will go from the GP41 to the CD4 positive macrophage cells over here. At that time this will pull these two molecules, this will pull these two cells towards each other. Whenever we get the pulling of this, the HIV and the CD4 positive macrophages over here, as a result we will see the fusions of these things, it will be easier. Okay, that means we can say this GP41 that will help to get fused, okay, along with the CD4 positive macrophage with the HIV virus, or the receptors, or you can say the molecule that is present within the, on the surface of the HIV. Okay, now whatever thing we'll get in terms of the CD4 positive T cells. Now this is all about the CD4 positive macrophage. Now what are the things we'll get in terms of the CD4 positive T cells? Okay, let's draw it again. It is the HIV. Within the HIV, as I said, we'll get the presence of GP41, and GP41 will get GP120. So this is the GP120. This is the GP41. Now, whenever they will be, try to understand, whenever you are talking in terms of the T helper cells over here, let's draw the T helper cells. These portions. Okay. Now is a CD4 positive T helper cell. Okay. Now, as we know that in these cases the CCR5, this chemokine receptor that is absent. So definitely there should be another kind of the receptors that can stimulate these things. Okay. So we'll talk about all these things right now. Now see, whenever we get the fusions, in the same way there is also presence of the what? No doubt there is the presence of the CD4 positive receptors. So whenever these receptors that will get bound along with the what? This is the CD4 or receptors that will get bound along with the GP120, glycoprotein 120. At that time again this will get the conformational changes within the glycoprotein 120, and it will stretch out again on more post-stimulations. So we'll get over here whatever the post-stimulatory things we'll get. Over here we'll get, it will come over here. For these things we'll get the another kind of the receptor that is present over here, where within the we'll get where, within the CD4 positive T cells. This is known as CXCR4. CXCR4. Now the CXCR4, this or receptor that is present where within the CD4 positive T cells. Now whenever the GP120 that will be activated after fusions along with the CD4 receptors, after that it will stretch out on more what? On more signals that will reach up to the CD4 positive TH cells or the T helper cells. And we will see within the TH cells there is a presence of the CXCR4 receptor that will get bound along with these things. And after that ultimately we will see the final signal from the glycoprotein 41 from here, it will reach over here, and that will pull the two cells, that means the, we can say over here the HIV virus and the CD4 positive T cells towards each other. As a result, we'll get the fusions that will be properly. Okay. Now we'll talk about that. Okay. In some pharmacological aspects and some population things. For example, if we see, if we get any kind of the mutations, for example, if we get any kind of the mutations within the CCR5, definitely this GP120 they will be not able to get bound along with these structures. Now this CCR5, if we the mutations within the CCR5 that can be found within the Europeans, North Europeans populations. Now within the North European populations we can get the mutations. We can get the homozygous mutations, or you can get the heterozygous mutations. Now if you get the homozygous mutations over here, definitely the GP120 that will be not able to get bound along with these structures. Or instead of that, if we get the heterozygous mutations over here, so for example the paternal genes, or you can say the maternal, out of the paternal and maternal, if we get one gene that is, that is having the mutations, another is normal over here, so definitely we get also the chances for fusions over here. But that fusions, that will take time, we will get the slower fusions over here. Okay, now if we get the mutations over here, so if you write it over here in North European people, okay, you can get the mutation within the CCR5. As a result, the glycoprotein 120, they will be not able to get bound along with these structures, CCR5. Okay. Now, on the same thing, if we see over here, that there are certain kinds of the pharmacological aspects. So there are some drugs that can bind along with the CCR5. So if we see kind of the drug, if we get any kind of the drug that can bind along with the what, CCR5. So whenever the drug these structures, let me show over here drugs. Drugs just like the Maraviroc, this drug. So let's talk about these things. Now the Maraviroc that is having the capacity to get bound along with the CCR5 receptor. So whenever the Maraviroc that will get bound along the CCR5 over here, as a result the glycoprotein 120 that will be not able to get fused along with these structures. So ultimately whenever the fusions will be not done properly, definitely the chances for the infection that will be reduced. So we can use the Maraviroc over here, and along with that other pharmacological agents we can use like the zidovudine. I will talk about all these things whenever we will keep on progressing about the pathogenesis or the pathophysiology. Okay, so just remember after this much that if we use the Maraviroc, so what is the function of the Maraviroc over here? The Maraviroc that is having the capacity to bound along with the CCR5. So this Maraviroc will get bound along with the CCR5 or receptor that is present within the macrophage or CD4 positive macrophage. So definitely the chances for the infection that will be less. It will be blocked totally. But another thing that till today that the scientists are trying to make the vaccines, but it's not possible to make the vaccines against the HIV. And the problem is over here. So if we write it over here, the vaccines, it's not possible. Why the vaccine can't make? For example, try to understand. If we make the antibodies, against the GP120, if we make the antibodies, write it over here. If we make the antibody against the glycoprotein 120, but this glycoprotein 120 is having the capacity to for within the glycoprotein 120 you will get one structures that is having the, or you can say that region that is known as hypervariable mutations. So within the glycoprotein 120 we will get a region, there we will get the hypervariable mutations. So whenever we are getting the hypervariable mutations, where within the, try to understand, within the glycoprotein 120 there are one portions where we will get the hypervariable mutations. So if we just keep on progressing or the development of the antibodies, but this this regions, that means on portions where the immune system that is supposed to work over there, but that region that will be mutated again and again. So if we just keep on progressing, if we just keep on trying to make the antibodies against the GP120, where actually the drugs they will get bound or the antibodies will get bound along these structures, but will get the hypervariable mutations within these structures. As a result, if we just try to make the vaccines or you can say antibodies over here, not perfectly matched with GP120 antigen. Okay, so whenever the antigens that is present in the glycoprotein 120, those antigen they are having the, I'm saying within the glycoprotein 120 one region, that there is the presence of one region that is having the hypervariable mutations, that is having the capacity to do the hypervariable mutations. So whenever we are getting the hypervariable mutations, we can make the antibodies, but whatever the problem will see, this antibody they will be not able to recognize the exact place where they are supposed to bind, because this GP120 that will keep on progressing in their hypervariable mutations. This type of the problem you can see over here. As a result, vaccination is not possible till today. So that's all about in terms of the HIV part. And in the second part, we'll talk about the next type of the pathogenesis. Basically, especially we'll talk about what type of the changes we'll get in terms of the RNA within the HIV, and what type of the outermost signals we'll get from here, and how and just prevent these things in the the pharmacological aspects, agents. We can talk about all these things in the next part. So this was all about, I said before only. There's the immune suppressions. You get the CNS disorders, the RNA retrovirus, or the reverse transcriptions. All these things, the normal things. Okay, I'm not repeating again. You can remember the Maravirok having the capacity to bound along with this one. So they will just inhibit the fusions. So whenever the fusions that will be inhibited, definitely we cannot get the infections within the normal T helpers, or you can say the normal macrophages over here. So that's all about in terms of the HIV pattern. In the next part, we'll talk about in details more about the pathophysiology. Okay.